Hello, welcome back to Learning Theatre. In today's video, we're going to talk about cell surface receptors. So, at the end of this video, you should be able to answer the following questions. Number one is, what are the three types of cell surface receptors? Number two, what are ion channel length receptors? Number three, what are enzyme length receptors? And number four, what are gene protein couple receptors? Now, what is cell surface receptors? Cell surface receptors, they are transmembrane integral proteins that interact with ligands and then initiate cellular response. And then the integral proteins that are used here means they are embedded in the cell membrane or the plasma membrane. So this one here is an example of integral protein. It crosses the cell membrane, so it is an integral protein because it cuts through the cell membrane. That's why we call it integral protein. And the first type of cell surface receptors is what we call the ion channel length receptors. Ion channel length receptors. As the name suggests, this receptor is linked to an ion channel protein that allows the movement of ions from the ions through the plasma membrane. So these ions here, these are the ions here, and then this is the channel. So when this ligand binds to this channel, the channel gets open so that these ions here can pass through successfully. And then so when it opens, this is how it happens. So this is the ligand here that has bind to the um, receptor. So when it binds to the receptor gets open. So when it's open like this, these are the ions passing through. And when the ions are done passing through, the receptor now, so like I was saying, this is the ion channel. This is the ion channel. And then when the ligand binds to it, the receptor gets open, and when the receptor gets open, the ions passes through the channel. And then when the ions are done passing through, the channel the receptor closes because the ligand that was attached will now detach from the receptor, and then when that happens, the channel closes back. Now let's talk about types of ion channels. Types of ion channels. Ion channels we are talking about is they are also known as ionotropic receptors, and then ion gated channels are pore forming memory channels, pore forming memory proteins that allows ions to pass through it. So this is because ions itself cannot move through the cell membrane or the plasma membrane, so they need a channel through which they can pass through. So these channels will help ions to get through the cell. So they will cross the membrane through these channels. And then the first type we have is what we call the ligand-gated ion channels. Ligand-gated ion channels. These are channels that allows ions to flow in and out of the cell in response to the binding of a ligand to the receptor. An example of this is what we call the acetylcholine receptors. So this is when the channel, it allows ions to flow in and out of the cell. So when the ligand bind to the channel, the ligand gated ion channel, when the ligand comes and then bind to the channel now opens so that the ions that wants to pass through will now pass through. And then that is why we call it ligand gated ion channels. It needs a ligand to bind to it in order for it to open, for ions to pass through. And then the second type we have is what we call the voltage gated ion channels. These channels involve the opening and then closing of ion channels in response to change in the memory potentials. So this voltage gated ion channel, This is also a channel that will only open when there is a change in the memory potentials. So let's say the external or the extracellular part of the cell was positive, plus, 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 and then the intracellular part of the cell was minus, minus, minus. So when it changes in voltage, the top now becomes minus, 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 and then the down becomes plus, 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 plus. And then when this happens, the channels, they get open, and then when the ligand has bind to them and then the memory potentials is changed, the channels get open so that the ions that wants to pass through them will now pass through them successfully. So we have there's in this type of channel we have a voltage sensor that is attached to the ion channel, and then change in this voltage will cause the opening of these ions to allow the movement of these channels, to allow the movement of ions. And then the voltage gated channel, we have so many of them. We have the ion, calcium, and chloride voltage gated channel, which allows the movement of calcium and chloride ions. Sorry, it's chloride ions, which allow the movement of chloride through this channel. And then we also have the sodium voltage gated ion, which also allow the movement of sodium ions. You have the calcium voltage gated ion, which allow the movement of calciums, and you also have the potassium voltage gated ion channels, which also allows the movement of potassium ions. Now, the third type of cell surface receptors is what we call the enzyme link receptors. Enzyme link receptors. They are called enzyme link receptors because they have an intrinsic enzymatic activity, or they are associated with an enzyme. And this enzyme is usually a kinase enzyme. So the most common one of this type of receptor is what we call the receptor tyrosine kinases. And then it has a specific phosphorylation of tyrosine residue, and then that is why it is called a tyrosine kinase, because of the tyrosine residue. And then the tyrosine kinase domain, which is this one, the one in the blue, that is the tyrosine kinase domain. And then this domain um, adds phosphate to the tyrosine residue, and then when it does that, ATP becomes ADP during this process. And then the function of this the kinase enzyme, is the addition of the phosphate. And then the second type of this enzyme link receptors is what we call the serine or threonine kinases. And then this one, the phosphorylation occurs in the hydroxyl side chains of serine or threonine amino acid. And then the third one, we also have what we call the tyrosine associated receptors. And then with this one, the tyrosine kinase, there is no tyrosine kinase domain, because there are certain enzymes that act through the cytoplasmic tyrosine kinases that is present in the cytoplasm. So this is our transmembrane domain, and then this is our tyrosine kinase domain in the color blue. Now, this kind of receptor with one receptor here is called a monomer. When there's only one receptor here, it is called a monomer. But when we have two of the receptors, we call it a dimer. And then so like this is example of the dimer. You see the receptors here are two. So we call this one a dimer. So fully activated tyrosine kinase receptor is known as a dimer. Fully activated tyrosine kinase receptor is known as a dimer. And then upon the binding of a ligand to the tyrosine kinase domain is activated. When the ligand binds to the tyrosine kinase domain, it gets activated, and then the monomers. So when they are activated, these two monomers will now come together to form what we call the dimer. And then at the domain of the second monomer, this one, the domain of the second monomer, which is this receptor, also get activated, and then get phosphorylated at the tyrosine residue. So in the tyrosine domain here, it will be activated and then get phosphorylated. Yes, so when there is only one, we call it a monomer. When there are two, we call it a dimer. And then the second the second tyrosine domain of the monomer will get activated and then phosphorylated. Now, the second type of cell surface receptors and the third type of cell surface receptors is what we call the gene protein couple receptors. Gene protein couple receptor. This is the most extensively studied receptors as it can respond to different types of signals or ligands. So it's also the largest family of signaling proteins that are targeted by 30 to 50% of drugs. And then this is integral membrane protein with seven hydrophobic transmembrane um, helices. And then they are connected by intracellular and extracellular loops. So this is the extracellular loops here connecting them together, and then the intracellular loops also here connecting them together. It is also called gene protein receptors because it is also associated with a protein complex known as a gene protein. And then it is a trimeric protein consisting of alpha, beta, and then gamma subunit. And then the alpha and beta subunit have small lipid molecules, helping them to anchor to the inner layer of the plasma membrane. And then if the alpha subunit is bound to GDP, then the receptor, if this alpha subunit is bound to the GDP here, then this receptor becomes inactive, or it is in inactive state or GDP bound state. So this is also known as what we call the off state. And on the binding of a ligand to the receptor, GDP is changed to GTP. And also the receptor becomes active or on state when all the subunit is bound to the GDP or GTP. And then this pro- protein complex, and this protein complex subunit is also known as T protein. Yeah, so in summary, today we'll talk about cell surface receptors, and we said we have three types of them. We have the ion channel link receptors, and then we also have the enzyme link receptors, and then the gene protein couple receptors. And then we said the first type, the ion channel link receptors. We have two types of them. We have the um, ligand gated ion channels, and then the voltage gated ion channels. Also, the enzyme link receptors, we said we have three types of them, which the first one is what we call the tyrosine kinase receptors, and then the second one is what we call the serine or threonine kinases, and then the last one is what we call the tyrosine kinase associated receptors. And then the last type of the cell surface receptors is what we call the gene protein couple receptors. Thank you for watching. Please like, subscribe, and then comment in the comment section. And also press the notification bell to be notified whenever we send a new video or we upload a new video. See you in our next video.